All right.
I wasn't what I did watch the lecture because by the grace of God, this man records the lecture.
All right, everybody. Hello. So we're gonna get going. How's your fall break? Good. Relaxing. Good. Hopefully you felt okay ignoring the homework that I assigned beforehand. I didn't mean to be like, hey, here's the homework. Enjoy your break, But fortunately, we've still got a week or so before the next exam, right? So,
Isn't it due November 1st?
Yeah, it should be November 1st because our exam's November 3rd, I think, right? So it'll be the class period before the exam, so we can go over that as a kind of exam review, check in on how things are going with that. So good. We finished up membrane stuff last time. I left off with a problem that I didn't solve. I kind of talked through it a little bit, so that's still in the slides if you want to go back to that. Welcome to it. So today I want to start getting into disinfection, which is sort of has been one of my focus topics for a lot of my graduate work and my research here. So it's definitely something that's close to home, and I like to, and I think maybe I mentioned this at the start of class, and I start getting a little confused sometimes when I just gave this lecture a week or two ago for the other class, and I'm just trying to remember what all I've soapboxed for you so far. So feel free to stop me if you're like, yeah, we've heard this. But I like to talk about my water bottle, which I inevitably neglect to toss in the dishwasher because I always forget it's like in my car or in my bag or something. So it's been a little while. So when you think about this, it's pretty clear that there's going to be some microorganisms in here. At least that's clear to me, right? Even though I filled it from the tap just this morning. So what's the difference between sterile, which this is not, and disinfected? That's kind of the question. So if I had a perfectly clean bottle and then I filled it up from the water fountain, do you think it would be sterile? Anybody? No? Okay, yeah. And I think I probably mentioned this already, but hopefully it's at least not infectious, right? So we would agree that hopefully it's not infectious. So even though when I take a drink of my water, I believe I'm going to ingest thousands, if not millions, of bacteria and viruses, even with that one gulp, it should not be infectious, right? Otherwise, I would have some trouble tonight, or whatever. So the idea here is disinfected really means it's not infectious any longer. So it's no pathogens at infectious doses. So we treat it to the point where it is no longer going to make people sick, or no longer going to have a condition where if you use it to cook with or something, that's going to become a problem in terms of pathogens. And that doesn't mean that there are no living pathogens in there necessarily. You might have deactivated the pathogens, maybe you've prevented them from replicating, in that way sterilizing them, so to speak. But so long as it's not infectious, we would call that sufficiently disinfected. Okay? Whereas sterile, like usually a little bit more geared towards use for laboratory settings or something where you really want to make sure there's nothing at all alive. Perhaps you also want to destroy any traces of DNA, or at least denature them with heat or something, to the point where you're not going to have any signal at all from any of your biological metrics or any of that. So sterilizing, you can think of like we often use an autoclave, let's say in my lab, but a steam cooker or a pressure cooker is essentially the same thing. And the object there is to completely destroy everything that could possibly live, microorganisms and all that, including fungus, whatever might be in there. And the way we do that is get a temperature above boiling point. So if you think of like a boil water advisory, the point of a boil water advisory is, hey, we had some issue with our water infrastructure, and now we're telling you to help disinfect your water because what we're giving to you might not be safe. We're worried it might be contaminated. So boil your water to achieve disinfection. It's not going to achieve sterility because you really need higher temperature than 100 degrees Celsius. This will be enough to disinfect, especially if you give it some time. But really to get high enough temperature, you have to have a pressurization system because typically you want about 121 Celsius to get sterility. And even that, you want to make sure everything in there is at 121 Celsius for at least 15, 20 minutes. So it's a little bit of, as we'll see later today, a contact time sort of a thing where you need those conditions for some amount of time to make sure everything is very thoroughly destroyed or transformed. So when we do a sterilization to do a disinfection experiment, for example, we want to make sure there's nothing else that's going to grow aside from the bacteria that we are interested in growing, or the viruses or whatever. So we want to sterilize it, and we do that with a big pressure cooker, essentially, commercial sized pressure cooker that's going to get steam to 121 Celsius, which is going to correspond to a certain amount of pressure. So it has to pressurize that and then let it cook for 15, 20 minutes before lowering the pressure back down and cooling it off. Okay, so the other thing about sterilizing, you could also consider a case, not this like autoclave completely, every protein has been cooked to no one's recollection. Any DNA is protein, so if you're going to cook the protein in that manner, so that's the one thing. But you can also sterilize in terms of causing them to not reproduce, right? So we have sterile animals. Either we sterilize them so that they can't reproduce, or we could even have hybrid animals, like a mule, for example, hybrid between a horse and a donkey, can't reproduce genetically, right? So that would be a sterile organism. So we could cause that perhaps with some, maybe ultraviolet light could denature some of the DNA, so they might be a functioning cell for a little while, but unable to reproduce, so therefore not infectious. Okay, so these two concepts are interesting, especially in that it might be okay if there's a few bad bacteria that we consume. Perhaps our stomachs can handle it or whatnot. So to really get into disinfection, we've now done all sorts of unit processes to remove particles, to clean up the water as best we can. Most of what we've done so far is particle removal. If you think about it, our sedimentation is particles. We do coagulation, flocculation to get smaller particles to settle by making them larger. And then we do filtration to capture even more. Sometimes we'll add here perhaps like a carbon filter, some sort of adsorbent. So sometimes this will include, as an option, activated carbon. And that will adsorb some chemicals. That's adsorb with a D. It's a surface phenomenon. Ad meaning it's adding to the surface rather than ab meaning it's like taking up within, like a sponge would absorb water. So sometimes we'll adsorb some chemicals. Sometimes we'll remove some metals or something like that. Technically, sometimes we'll add in a water softening component here. That's more chemistry than I will terrorize you with, so I will refrain from doing so. But if you're curious, that'll be going on in my other class in the next few lectures. So feel free to tune in for extra content at 9 a.m. Eastern. Okay.
Like a bot on Twitch.
Yeah, same thing. Yeah. So if you legitimately, if you are curious, it'll be on Twitch, and then you could find my other playlist on YouTube, and it'll be there too. So feel free. And water softening is kind of interesting. It's just a lot of equilibrium chemistry. And I don't want to, do that to all of you. Okay, so for the disinfection process, then we've cleaned up the water a fair bit, and now we're ready to disinfect because the bacteria, the viruses, and everything no longer have particles to hide behind, essentially, right? So we want to make sure that the chlorine or whatever disinfectant we're using is going to have the highest impact, so we don't over-chlorinate, so we don't have to spend as much money on the chlorine. And we'll see several reasons that we want a nice, clean water to optimize our disinfectant. From there, sometimes we'll add fluoride. It's a dental health thing, and it's sometimes a tracer test to see how well the water is moving through the pipes. It's not very reactive with much of anything, although it does tend to help tooth enamel have kind of a stronger protection. And that distribution system itself, usually we send it out with enough residual chlorine in a what we would call a combined chlorine. So we want a little bit of what we call chloramines because they're not very reactive, but they'll be reactive enough to prevent any bacteria that was damaged but not killed all the way from repairing themselves and becoming happy. So it's a way we keep the distribution system relatively clean, or at least cleaner, without having so much harsh chlorine that it would erode the pipes, right? Because chlorine is an oxidant, and rust is essentially oxidized metal, right? So we don't want to oxidize all the pipes. So by design, we send a small amount of chlorine, hopefully not too oxidizing conditions there for the chlorine to go through. Okay, so that kind of brings us to how do we know how much disinfection is needed? And this is always a very entertaining thought. If you had to figure out how many noroviruses it takes to get somebody sick, what would you do? How would you do that experiment? Someone's going to get sick. In fact, the CDC apparently, I learned this, I took a microbiology or an environmental microbiology class at Emory's public health school. I was cross-enrolled just kind of for fun and to get that the health perspective side of it. And it was a very entertaining class on food and waterborne diseases. And I learned that the CDC literally, and I don't know if they still do this, but at some point they were offering like $1,000 to young, healthy volunteers to come and have like a hotel type environment to sit there and you take a pill and you'd just be hoping that you took the placebo or that you were an asymptomatic case, because you didn't know if it was like one virus, if it was 200 viruses, if it was just sugar, no virus. You didn't know what you got, but they were going to feed you and control what food you got, so they made sure that the food.
How do they ensure that it stays alive with the capsule?
That's a good question. I'll get back to that in a second. So you basically have this environment where they're making sure you don't ingest any viruses from what you eat or anything else, and then you sit there and you may or may not get sick. So they'll monitor you for a week to make sure you're in good health, or if you're not so good health, they'll take care of you. But and then they'll give you $1,000 for doing this, right? So it's like, would I risk norovirus for for $1,000? And I'm thinking, as a grad student, college student, I could just sit there and study or play computer games or whatever. It's like in terms of keeping the virus alive, it's not too difficult to get a protective environment for bacteria or a virus like that. Normally you'd keep it kind of probably in a deep freezer, but then to administer it, it'd be kind of a little bit like a vaccine or something where you don't want to let it sit at room temperature for a long time. You probably want, if it's a virus, you probably want to keep it in a moist environment so it doesn't sort of dry out and disintegrate, as most viruses do. But if you do that, you could probably just put it in a pill. And then the way you would actually determine how many viruses you're giving somebody is typically by serially diluting. So you take like high concentrated stock that you know something, it's maybe like 10 million viruses in there. You dilute it by a factor of 10, so then it's 1 million, another factor of 10, and you keep going and you use your best estimate. So you can get pretty accurate by diluting like 1 in 10 repeatedly. That's a serial dilution. We do that all the time in kind of disinfection studies. So again, that's sort of how many logs did you dilute it, right? It's like a 5 log dilution would be going from 1 million to 10, right? Because you diluted it 5 times. You're just basically moving 1 digit each time. So then you give that and say, okay, we think that's approximately 5 viruses that you just ingested, or likely to be around 5 viruses. And so it's kind of crazy, but that is literally how it worked. And so they've discovered that things like norovirus and rotavirus, they're pretty infectious. It only takes, I think it was something like tens of viruses to get somebody sick. Of course, some lucky fellows would be asymptomatic, so they might have the virus, it might be there, but it's not giving them symptoms. I think poliovirus might have been a little bit higher, but so maybe hundreds. I can't actually recall too well. Vibrio cholera is an interesting one. This one actually requires something like 10 to the 6th to 10 to the 7th, so 1 million to 10 million bacteria cells to get somebody sick. And that's very interesting to know. The thing is, it's vulnerable to stomach acid. So acidic conditions kills that particular bacteria quite quickly. That's a good thing, because when it gets out into the environment, somebody excretes it from being sick. It tends to be excreted in very, very high volume, high amounts, high numbers, because it kind of needs that to get its next host infected. And so something like raw oysters that do a great job grabbing stuff out of the water, cholera is a significant concern there, because it will stick to the fleshy parts of the oyster and stay there. So if you eat it raw, you're at some risk. So if you do, I suggest eating it on an empty stomach. Would be my best advice, or just don't do it. Likewise, noroviruses are also a known issue with raw oysters sometimes. And it really is about that fecal oral route of transmission, right? Did somebody who was sick discharge waste into a wastewater treatment plant, and that went through eventually discharged into the river and did not, was not disinfected fully or whatever, and then that water made its way by this oyster bed. It's some gambles there, and there's a lot of advice out there about what season or what location or something, probably all somewhat relevant to where's the closest wastewater treatment system and how well is it disinfecting its effluent.
When you put lemon on an oyster, would that kind of kill the Vibrio?
Yeah, so lemon is a great way. In fact, ceviche is essentially you're cooking it in kind of citric acid type stuff, right? The lemon or the lime, and it is quite effective at a lot of things. So certainly for Vibrio, that would be a good way to make it safe. Give it a little bit of time to be there. If you just splash it on and consume right away, maybe you're relying more on your stomach acid to give it that time. But if you were to let it sit there for some number of minutes or something, that would probably be a lot safer than just straight up eating it. And that should be effective against a lot of pathogens. So ceviche, I actually don't know the details about if there's some risk. There might be some organisms that form like a cyst. Some of these, the Cryptosporidium, for example, and some amoeba form a kind of a environmentally resistant cyst. They can resist chlorine and stuff like that. So some things might still be there. Wouldn't be like a guarantee like heat would so much, but could be a good strategy. E. coli and Salmonella tend to be something like 10 to the 4th, so tens of thousands, which is again why if there were 100 pathogenic E. coli in here, I might like, if I just happen to know that, I might be like, yeah, maybe I should go and clean it. But I also wouldn't be too worried. It's very unlikely that even if I drank this whole thing, that would make me sick.
I don't know. I'm assuming that these have a chance to reproduce and whatnot.
Well, that's these studies were a way to give them that chance, right? So some of them are going to be damaged in my stomach acid, right, presumably. And if there's some if there's like one bacterial cell and it starts reproducing, it might be that my natural immunities or other bacteria or other organisms are like fighting for the food and stuff. It might just simply, on average, not become an infection because it was dealt with or otherwise outcompeted or something in time. Maybe it could, and which is why if I were to be given that sudden insight, I'd be like, maybe I'll just leave that be, pour it out, and clean it properly, right? Obviously I'm not suspecting that would be an issue, but so it's kind of an interesting thing. It's like how many does it take, and how do we know? For Cryptosporidium, it really only takes like 2 to 5 cells. So that one, if you get one, you're in trouble. So, and that these ones, so the classes of bacteria play a role, or classes of organisms here have a role in this. So viruses are pretty much like very vulnerable. Some of them have a little bit of an envelope that protects them a little bit, but it's still like just raw proteins and maybe a little bit of a lipid barrier, but very vulnerable to the environment, very vulnerable to disinfectants. Generally, bacteria at least have this, what's called a phospholipid bilayer. It's just some sort of like a fatty capsule to keep them a little bit protected. Whereas, and if you remember back from biology, these would be prokaryotes. How we spell it? Prokaryotes. And then protozoa here would be eukaryotes, meaning they have a full cell wall. So they're true cells, right? The EU, I think, is true or whatever. So it's like these single cell organisms that have like your standard like animal cell type of cell. And that gives them more protection inherently. And then they're sort of like more sophisticated organisms, tend to be bigger as well. So usually these are like, 1 to 10 micrometers, whereas bacteria are usually like 0.5 to 1 micrometer. There are some weird exceptions, but like some even large enough that maybe you could see with your visible eye if you collected them. But at any rate, essentially the dose makes the poison, right? And so what we have to figure out as water treatment experts is how many are there, how many do we expect might be in the water, and therefore what kind of treatment do we have to apply to make sure that it's safe. Technically disinfected, meaning nobody's going to get sick from it. Okay, so that's...
What does the SPP stand for again?
Different species of Giardia. So that's like this is the family, and then there's several genus names that are, or genus and species. Sorry, this is the genus, Giardia is genus, SPP is species in that genus. So whereas Cryptosporidium parvum, this is the species, Cryptosporidium is the genus, Giardia is another class of organisms, and there are several different species that can make you sick. It's like Escherichia coli. There might be other types of Escherichia bacteria, like Salmonella species. There's several of those. One thing I didn't mention actually is some bacteria will create toxins, and this might be their mode of making you sick, is creating a toxin inside of you. That compound itself is toxic. Some others might create toxins in the food than you eat later. So actually there's a big difference between food poisoning and food infection. And most of the time we always say food poisoning, but usually that's a food infection. So a food infection is, or water, could be water too, but if we're talking about food poisoning, we're talking about like vernacularly, the way we normally use it is like, oh, I ate something at that sketchy restaurant and that night I got sick, right? It's like 8 hours, 10 hours later. And by the way, the other thing they studied with all of these is like, okay, how long until the onset of illness, right? How long did it take until you get sick? And norovirus is notoriously like 6 to 8 hours. So usually if you had something for dinner and then got sick 2 hours later, it's probably actually lunch that did it, but there's something called recency bias. Your brain's going to think to yourself, oh man, that pizza was terrible, but maybe it was the salad you ate for lunch or something. So it's kind of a little interesting factoid. Now, that's food infection if you're having an infection of organisms growing in you causing you to be sick. That's food infection. Food poisoning is literally something created a toxin, or maybe somebody poisoned your food. And then it's pretty much immediate. As soon as it's in you and your body's like starting to react with the poison, you become violently ill, typically, depending on what it is. Probably the most dangerous and well-known would be botulism poisoning, or botulinum. So there's a bacteria that is usually not such a bad thing, and your stomach acid kind of neutralizes it, typically with no problem. But if it happens to be growing in your food and given plenty of time, it could create a lot of botulinum toxin, which is a very potent neurotoxin, which is why we use it. Botox to remove wrinkles is basically deadening any of those nerves that were causing your skin muscles to like tighten a little bit and create wrinkles, right? So it's just like if you're using Botox, it's injecting this really potent toxin on purpose, applied carefully, so that it neutralizes just some certain nerves, right? But if you ingest it, you probably die of paralysis pretty quickly, because then your whole body's sort of shutting down. As it relates to stomach acid, the reason you are not supposed to feed honey, raw honey, to infants is because it often contains the botulinum bacteria. I forget the exact name here, and the thing is, infants don't have a very acidic stomach environment. And so up until a certain age, those bacteria can then potentially start making a living in the upper stomach, like in the stomach, or maybe it's the upper intestine, and start producing the toxin, which is then a very bad thing. So as adults, or even, I think even as early as maybe 2 or 3 years old, it's no problem. Because the stomach acid just does so much for us. Okay, so a little bit of history. And here I'm highlighting Jon Snow, not my brother and not the Game of Thrones dude, but this old guy who, and I'm not going to ask you who I resemble most, but I will say that the old dude here did not have any direct descendants himself. So if I'm related, then it must be one of his siblings or something. I don't know. Okay, but we call Jon Snow here the Father of Epidemiology because he was one of the first to identify truly the source of a disease outbreak and map it and figure it out based on mapping who's getting sick and basically establishing a method to learn about disease based on rates of disease. And that is kind of what epidemiology is. And he's famous for an 1850s cholera outbreak in London. If you've ever been or heard of it, that's the Broad Street Pump. This was the deal there. And really, this was kind of one of the first confirmations of the germ theory of disease, which is a very important concept. And if you think back about like people dying and getting disease without having a clue what it's coming from, that would be freaky. Like if you just had no idea why you're sick, but you're sick, and why did your whole family just get sick? But it's like, what is this? Magic or something?
Yeah,
like what would you?
Didn't they think it was like particles or something in the air? I forget what they call
Yeah,
it.
miasma.
Yeah.
Miasma theory. So that was one of the things that he was dealing with and kind of pushing back against. So he created what's called the ghost map. This is also the title of a really an easy and interesting read. So I highly encourage you to take a look if you get the chance. This is essentially a fictionalized recounting of Jon Snow's discovery of the source of this cholera outbreak in London. So fictionalized meaning they gave everybody, they put words in everybody's mouths. They created fictitious conversations as if it was happening. So you can read it like a real story. But it holds true to the detail of what happened, right? So they're essentially just making a historical account, but what people literally said was never recorded or whatever. So they're adding that to make it story form. So this map over here, on the right, this is a section of London that was plagued by this outbreak in, I think it was 1854. And this is a cholera outbreak. So I mentioned cholera earlier, Vibrio cholera. It takes a whole lot of them to get you sick. Well, the terrifying thing about cholera is if you don't know how to deal with it, it is lethal within hours sometimes. So you might be happy and healthy in the morning and dead in the evening. It is possible. The deal is cholera does is once it infects somebody's intestines, is essentially our intestines normally are extracting water out of whatever is inside the stomach, the food and all that. It's pulling water out from what we've ingested into our bloodstream. Cholera takes command of those cells and reverses the order. So it causes water to be exchanged in reverse. So it's extracting water from our bloodstream and discharging it into our intestines. Obviously, that gives terrible diarrhea that's like super watery and all that. But that's, if you from a disease perspective, that's horrifying in terms of how quickly that will dehydrate you and therefore kill you. Now, the fortunate thing today is we know how to deal with dehydration. If you basically have access to very basic healthcare, an IV easily save your life. You probably have no issues. You'll just be very uncomfortable for a couple of days. Right? You have what's called a cholera cot, so you can lay there, be sick, and be fine, assuming you have hydration. Even hydration salts. You just have Gatorade, for example, would probably be enough to make you have no issues, because that hydration, you really, what the big risk there is, you can't just keep drinking water because that's going to pull all the salts out as well, and the salts help you keep the water balance. So you need salt with the water for severe cases of dehydration like this. So anyway, at the time in the 1850s, you would have an outbreak like this, absolutely terrifying, people dropping dead everywhere, and almost overnight, right? So then what he did, and this is kind of morbid detail here, but he made a map of these little black bars at different people's houses, signifying who died, or how many people died in each house. So there's a better rendition of that, kind of three-dimensionalized a little bit, and red bars here to help them stand out. And you see like this map here that had that big number there. They've just turned that and said that's this peak here. So many people dying, many people in the same house even. And so what Jon Snow did was he went around to try to figure out what was going on, try to figure out, okay, where is this happening? Why is it crossing different neighborhood boundaries, and like really trying to test these theories of like this miasma, the gross odor, vapor stuff. And you can imagine how awful the urban city in the 1850s with very poor sanitation would be. It was not a pleasant place, and so there was, understandably a lot of thoughts about, okay, just the grossness hanging in the air is giving people sickness. So once he made this map, he also then kind of pinpointed each of the wellheads. So there was different wells in the neighborhoods, pumps to pull water out of the ground. So he used groundwater, and he noticed that really everything was centered around this Broad Street pump right there. And so he decided to take the handle off the pump, force people to get their water from somewhere else. And as soon as he did, the outbreak kind of petered off and stopped. So ultimately he recognized that this was a waterborne thing. He thought he saw something in a water sample he took. Probably was wrong, because he probably didn't have truly the technology to see small enough to see bacteria. But the idea was there, and he connected that and was accidentally right, perhaps. And it turns out that they learned that right above this Broad Street pump, there was a house that had a sick infant. And the infant, because it was an infant, was not as affected by cholera. I I guess that, since the stomach's not quite as efficient at things, the disease just did not affect it so badly, and it lived at least for a few weeks, and it might have even survived. But the problem was it was discharging its waste into their little latrine system, which happened to drain basically straight into the well, which is pretty awful, right? So you kind of realize, okay, well, none of these might be good ones to drink from, but the fact that there was that continuous source of cholera straight into the drinking water for this area was the issue. And apparently that infant had traveled with the parents and recently come back, and they brought the sickness back from somewhere else. Is kind of how all of that came together. So kind of a crazy story. That's why we call Jon Snow the father of epidemiology, way before Game of Thrones popularized the imposter. Okay, so quite fascinating. Definitely encourage you to take a look at that morbid book. It is a good read. It's a interesting, engaging, if very dark. Okay, so disinfection strategies then. How do we kill these nasty bacteria, viruses, and all that? Chlorine is kind of one of the obvious ones. So free chlorine is going to be probably the most common, and we can apply it in a couple different forms. We could do Cl2 gas. So if we take chlorine gas and react that with water, what we're going to get is HClO, essentially, or HOCl. We could rewrite it. So we're going to take the HO from the water, add the Cl here. That'll leave us with one hydrogen remaining, so it's going to acidify the water a little bit. And that's going to mean we have one Cl minus free out in solution. So that's one way to get our HOCl. That's what we're going to call free chlorine, is the combination of HOCl and OCl minus. And if we remember, I think we've talked about this once or twice already, HOCl is going to be in equilibrium with OCl minus and H plus. So whenever we have HOCl, we're going to have some OCl minus and vice versa. So those two are going to exist together. The total together is going to be what we call free chlorine. So in terms of conservation of mass or number here, we can look at free chlorine as a conservation of that OCl group, whether it's OCl minus or HOCl. That the total amount is going to be the total free chlorine. The other way we can apply this is simply as NaOCl or maybe CaOCl2, and that's going to be essentially adding it like this. NaOCl would then result in OCl minus plus Na plus. Okay, and we could do it with calcium hypochlorite, and we have a calcium CaOCl. With two of those, we'll get two OCl minus to balance out the one calcium that has a 2 plus charge. So could write that like so. So it doesn't really matter which way we apply it. One thing to remember here is that we have these forward reactions. So if we're going to add NaOCl, it's all going to Na plus and OCl minus. It's a dissociation. It's going to dissolve. It's just going to be separated in water. We're not going backwards to reform NaOCl. It's just all going to Na plus ion and OCl minus. Then the OCl minus will be in equilibrium, right? So we could go from there then into this, and we'll be back and forth with HOCl and OCl minus. Does that make sense? All right. So that means for our free chlorine, pH is always going to be important because it's always a part of that equilibrium. And it turns out HOCl is a stronger disinfectant by a long shot, so we really do care. We need to have mostly the HOCl for effective chlorination. We could also apply chlorine in terms of chlorine dioxide. Still is a very oxidizing agent. There's probably chemical reaction pathways that can bring us back to free chlorine. I think this would also be technically considered as a kind of free chlorine. We don't, it's kind of a separate system. I'm not going to bother going too much into that. And I actually don't even know a whole lot about the chemistry there, aside from that it can act directly as a disinfectant. So then we have combined chlorine. If the free chlorine that we're typically using encounters some sort of amine or ammonia group in the water, a lot of times that will cause a reaction so that you form what's called a chloramine. So instead of just NH3, we're going to end up getting NH2Cl or NHCl2 for dichloramine, or NCl3 for trichloramine. So we're going to have some form of the chlorines replacing the hydrogens here, and these compounds are going to be slower acting disinfectants. So this is what I said, we often intentionally add some chloramines to go keep our distribution systems a little bit cleaner because they act slowly, they're not too harsh on the pipe systems, and we can better make sure that some of the chlorine makes it all the way into the tap so that it kind of keeps everything a little cleaner. Now, one thing to note here is if you've ever been swimming in a swimming pool and been like, oh, wow, that's really irritating my eyes. The chlorine is so strong. Smells so much like chlorine. Really what you're smelling and feeling is most likely di- and trichloramines. That means there's been a lot of ammonia, a lot of nitrogen in the water, which means either there's been a lot of people sweating in the water or urinating in the water, most likely. Now, maybe, let's just put this out here, maybe whatever water source was using had a fair bit of ammonia in it, or like nitrogen, maybe there's something converting from nitrates that naturally occurring into ammonia. Generally, it's just yucky, right? And it's unpleasant too. So hopefully that means there's enough chlorine in there that it's microbiologically relatively safe, but it's just no fun, right? So sorry to ruin your future swimming experiences, but that's actually what's happening, because these two are a little bit more volatile and much more irritating and smellier, and give that sharp, acrid chlorine smell. All right, so another chemical disinfectant would be ozone, so O3. Another oxygen molecule. Typically when we talk about oxygen or molecular oxygen, we're talking about O2, the stuff we breathe. Well, we don't love to breathe O3 because it's a lung irritant, natural oxidant. It's just less stable and still, as the name implies, likes to oxidize things, right? That means it likes to take electrons away from stuff. It's an oxidation reaction. We could use direct UV light to cause disinfection. We'll talk about that in a little more detail, probably next time, probably next time. So in terms of chemicals, those four are the primary ones. Sometimes we could use what we'd call an advanced oxidation technique, where we combine chemicals or chemicals plus UV in such a manner that we cause highly, highly reactive radicals to be created. So, for example, if we have hydrogen peroxide, we can split that with UV and create essentially hydroxyl radicals, which are extremely reactive, so reactive that they only live for nanoseconds. They'll react with water, they'll react with basically anything, they'll react with each other. They're going to find something to react with and do so. So they're good at destroying pharmaceuticals, things like that. But if you have natural organic matter in your water, it's going to react with that. So you might get some wasted energy there. Hydrogen peroxide also reacts very strongly with iron. It's Fenton chemistry, also producing hydroxyl radicals. So if you've ever had a little scrape and wanted to disinfect it, and you poured hydrogen peroxide and it gets all fizzy, that's the peroxide reacting with the iron in your blood. And you'll notice if there's no blood, you're not really going to have much fizz. So it's really that reaction with the iron in the blood creating really, really strong oxidants, which presumably will do a pretty good job killing any bacteria that might otherwise infect you. You don't want to do it overboard because then you're going to be oxidizing your cells and stuff that are already damaged and wanting to repair. So you wouldn't want to like soak it there for hours and hours by any means, but pretty effective for a quick way to clean a wound. Okay, so free chlorine. Mentioned earlier, HOCl is the stronger one, so we want to make sure that we are biased towards HOCl. Remembering that HOCl and OCl minus are in this equilibrium. And what that means is if we were to track the fraction of total chlorine as a plot, let's say where 1 is 100% one side or the other, versus pH, and let's say we go all the way from 0 to 14 here, we'll call this 7. Well, we know that our pKa is usually about 7.54. It is a little bit temperature dependent, so you'll see some problems give you a temperature. We're not going to deal with that. I'm just going to give you the value. And this is coming from that equilibrium constant 10 to the negative 7.54. If you recall, the p meaning we take the negative log of, if we take the negative log of 10 to the negative 7.54, that's a pKa 7.54. So what this means is that's the point at which they're going to be equal. Recalling the fact that Ka is that products over reactants. And that would look like H plus times OCl minus the molar concentrations here divided by HOCl. So this is where we get the equation, right? That's our definition. So the point at which HOCl and OCl minus are equal is when the H plus is equal to Ka. So let me elaborate. I'm just going to take this and simplify and say OCl minus divided by HOCl. We can solve for just that ratio and say that's going to be equal to Ka over H plus. And so if I were to ask you, when is HOCl equal to OCl minus? Well, if you look at this, you could answer, well, if you set those two equal, that's going to be 1 on the left, right? So 1 equals Ka over H plus. That means that H plus is equal to Ka when the left side is 1, right? So that means Ka is equal to this H plus at that condition. So when we set that equal to 1, then that happens, right? So it's simple algebra, but I'm taking some time to explain it so that we can see that that's how we can understand this equilibrium moment here. So at pH 7.54, these two are going to be right at the halfway mark, right? The 0.5. They're going to be equal to each other right there. Okay, so from there, if I take the HOCl side, under acidic conditions, it's more likely that we add the H onto the molecule. So we're pushing this reaction left when we have more H plus, it pushes that reaction left. And so that means we have almost all the way HOCl for acidic conditions, and then it starts going down and then drops to close to 0 by the time we're at 14, pH 14. On the reverse side, we have almost everything's as OCl minus way up at basic pHs, and it's going to kind of do the same thing where we cross at that point and we pretty much approach 0 by the time we're very acidic. I didn't draw it perfectly, but you get the picture here that they intersect at that point where pH is 7.54. Okay, so that's a good way to take a look and say, just even estimate, okay, if we're at pH 6, then, hey, that's going to be something like 70 to 80, maybe 90% of that is going to be HOCl, right? Whereas if we're at pH 8, we're going to have more OCl minus. Maybe 60, 70% of it is going to be OCl minus. Okay, so that means that during water treatment operations, we generally try to get the pH between 5 and 6. It doesn't take a lot of acid to change it that way, that amount, but we get a great impact in terms of how much of our free chlorine is HOCl. And if it really is something like 10 times stronger than OCl minus, that is a really good benefit, even if we have to adjust the pH back later. It tends to be a relatively straightforward process. Okay, so I'll give you a problem then to think about, and I'll probably take the first couple steps to help you along the way. If we've got 15 milligrams per liter of HOCl added to a potable water for disinfection, the final measured pH is 7. What percent of HOCl is not dissociated? It says assume the temperature is 25 degrees Celsius, and we'll just say that means Ka is 10 to the negative 7.54. Okay, so it's asking what percent of HOCl is not dissociated. The dissociation reaction is HOCl dissociating into OCl minus and H plus. We know that some of it also returns, but the dissociation is just saying it's going to break apart. So what percent of it, essentially when it says is not dissociated, means what percent is still in this form. Okay, so at equilibrium at pH 7. So I'll join you in a couple minutes to give you a couple steps, but take a minute, see how you would approach that.
pH is 7, 7.54. Or did you say that right? Oh, sorry. I just didn't catch it at all.
What's that? The pH is 7.
So therefore, Ka.
So
When
the
it's due to the negative 7. No, that is because of that. Yeah. Okay.
So at pH 7, we know that H plus is equal to 10 to the minus 7.
Oh, figure out that. No, I didn't do math. But it's not that.
No, that's you.
I feel like we might have gone with that. I think we might be able to find it, so. Let's see if I can concentration by concentration or scale percentage. It's all the same. Part of it all. It's still somewhat of a. But we still need to get some. Yes. Let me ask. Right, so you're gonna say percentage points for. I'm pretty sure this was the way. Why did we do that? That's good. I thought we were forgetting something. Yeah. Well, there's no weird spot. No red herring and stuff.
All right, so I'm gonna jump in here and kind of walk you through this. It's just hearing no red herring here. So this problem says we got 15 milligrams per liter of HOCl added, and then we presume from there it's going to dissociate or, it's going to go through the equilibrium reaction. So it's not going to be the full 15 milligrams per liter that is staying as HOCl. The question would have to be much more clear about that, if so. But even if we just put that aside for a second and then continue reading, it says the measured pH is 7, and then what percent of the HOCl is not dissociated. The percent, given some pH, the percent that's going to be one or the other is actually not relevant, not related to the amount we put in, right? So if we had a problem that was then asking us to say, okay, how much disinfection do we achieve? Then we need to know exactly how much HOCl we have. Then this 15 milligrams per liter becomes very relevant. So if this question continued and asked us that, and I have given problems like that on exams before, then we need to definitely go into the molecular weight, have the molecular weight, we convert from that mass to molar so that we can take whatever ratio we find here and solve that way. Now you could use it and solve for an exact value for HOCl, and then you can solve for an exact value of HOCl minus and do the math on the exact values, but that should all come back to the basic equilibrium relationship that we describe essentially with this Ka equation. So what we're going to do, or what we'll see, is anytime you have an equilibrium like this, the Ka equation can be used to give us information about the ratio, right? We have this ratio of HOCl to OCl minus, just like we did a minute ago when we said, okay, at what time is the ratio equal to 1, or at what concentration of H plus, or is that ratio equal to 1? Here, we can ask ourselves, what is the ratio of OCl minus to HOCl when the Ka to H plus ratio is not 1, right? So we could do this by saying Ka over H plus and put in these numbers as 10 to the negative 7.54 divided by 10 to the minus 7 gives us some information then about our ratio between OCl minus and HOCl. So we could solve that and say, okay, with given this ratio, then we find we could probably just take that and do algebra to find for ourselves the fraction that is the total. So given that we have some ratio of one to the other, we could maybe set the one value to 1 and then look at the comparison. Another way we could do it is to consider, if we write it out a little more elaborately, the ratio that we're looking for is HOCl divided by HOCl plus OCl minus. That's the part in the total. And if we can get this part in the total in terms of this direct ratio of OCl minus to HOCl, then it becomes a very simple substitution. Now, it doesn't really matter which way you go. You're going to end up doing the same algebra, it just maybe at different steps. So